Welcome to Superheroes of Science. I'm Stephen.
And I'm Sarah. We co-host Science from the Experts. Our guests are professionals doing cutting-edge science right now.
They're experts in their field discussing what they know best. So listen up and learn real science from real people.
Subscribe now and stay informed of our latest episodes and show your support.
Well, welcome to another episode of Superheroes of Science, where we are here with Imrul Shahriar, who is here at Purdue University researching in the field of drug development.
Thank you so much for inviting me. I'm very glad to be here.
Yeah.
Oh, we appreciate you taking time because the research you're doing is extremely important. And obviously to everyone in the world.
Right.
So that's a big population. That's good. Yeah. But when we say drug discovery, what do you mean by that? What is it that you're researching? I know that I asked you earlier beforehand, it's like, Mike, are you like an organic chemist? What are you really? And you're like, well, I do biology and chemistry, and you can't really define and put. You refuse to not be put you in a box,
Yeah.
which is probably safe.
Yeah.
But so what is, explain drug discovery. What does a scientist like yourself do in the field of drug discovery?
Okay, so I have to say that drug discovery is an interdisciplinary field. So it's a kind of a borderless science. There's no border. So we can't define drug discovery by physics, chemistry, biology, or any other particular field. Drug discovery encompasses all the areas of science, I would say. So, for example, when you want to discover a drug, you have to find a disease that you want to treat. And for that particular disease, you find a protein. So a protein that is a biomolecule, so that you want to target. So if I target that protein with a small molecule or a peptide or something that can modulate the function of that protein. So that's your first step. So to identify a target, so that you want to shoot at. So that's the first step. So to identify a target, you need to have a lot of understanding of biology. So how the biology of that particular protein actually works. And then the next step is if you are interested in small molecules, so the chemist that we are interested in, so I'm trained as a chemist. My undergrad and my Ph.D. is also in chemistry and in the organic division.
Yes.
Okay.
Yes, I feel justified right now.
Okay, so we want to find small molecules that can bind to that protein and selectively. So we don't want that molecule to bind to everything in your body because your body has a lot of proteins. So we want to find a molecule that binds selectively only to that particular protein that we want to target. So it's a very selective one. And for that, we can do screening. So we can use computational tools. So to do in silico in our computers, so we can screen against those proteins. And also then we can, after we identify some promising candidates, so you start, for example, with a library of millions, and then you narrow it down to maybe 10 or 20 molecules that you want to test in the lab. Then you can, you do some in vitro testing, so in the lab, and finally we can take it to in vivo, so to test in mice and or in any other animal models. So in the whole drug discovery process, when you identify a molecule, you have to synthesize it. So there comes the chemistry. So you need to find an efficient way to make that molecule so that it has got high yield and all the good properties, characterizations. So there the chemistry is involved. And when you are testing it in, for example, in human cell lines or in vitro or in animal models, there comes a lot of biology. You have to learn how to handle mice, how to do injections. So there you are also involved in some sort of basic medical science or animal procedures, and you get to do surgeries and collect lungs and measure virus titers. So many things. So starting from identification of a target to testing it in animal models, so that ultimately we can take it into human clinical trials. So that's the whole path of drug discovery.
You make it sound so simple, and it's not.
Yeah, it's a very lengthy process, of course. Yeah.
Now, refresh my memory a little bit. I know people have told me before, but it's obviously I don't remember much. When you say a protein, what? And I know there's all kinds of proteins in the body and stuff. Now, why is it we're trying to bond to a protein?
Okay, so protein is a biomolecule, so it's a natural polymer. it's a polymer of amino acids that we call. So amino acids, lot of amino acids, they come together and form a protein. And
Okay.
this protein does everything in your body. It can transport molecules from outside of the cells to inside. It can transport ions. It can do so many functions. It can induce some signaling that is important for a particular function in your body. For example, your metabolism, or to fight against diseases, to activate your immune system. For example, how your immune system, so immune system is our, like the policing system in our body. So whenever we have an infection from something outside, like a virus or a bacteria, then that immune system gets activated. So how the immune system actually knows? Through some proteins. So proteins play all the role in identification of whether there's any foreign thing is in your body or not.
Right.
Yeah. So proteins do everything in our body. Yeah.
Okay.
Yeah.
Now, what is it you're researching now?
Okay. Currently, our focus is on influenza. So we are trying to develop a new drug against influenza infections. So because influenza, we know that there's a vaccine, we get flu shot every year, but the vaccine coverage is not that high, and the vaccine effectiveness is not that great. And the antiviral drugs we have currently has also limited efficacy. So you have to take them within 48 hours of getting flu symptoms. So to make that work effectively. But if you are at a later stage, then it would not. It won't work as expected. So there's a limitation, and all the efficacy also is different because it only will reduce your symptoms by 1 or 2 days. So that is not great. We want a rapid recovery from viral infection. So there we thought actually how we can specifically target the virus-infected cells and the virus particles. So we wanted to identify a protein that is expressed only on the infected cells and not on your healthy cells in your body.
Okay.
So that's a targeted approach. So that actually what our lab does in Dr. Lau's lab. We try to do targeted therapies. So for example, we work on a lot of cancers. So we want to find a protein that is expressed only on the cancer cells and not on your healthy cells.
Oh.
So that you can directly target the cancer cells and kill them. So in case of virus infection, we do the same. So we target the virus-infected cells specifically and the virus particles and nothing else. So for that, you need to find a protein. And then what we do is we actually take the advantage of our immune system. So we have another, so we actually develop a small molecule that can find and locate where your infections are in your body, and then call in your immune system. Hey, there's an infected cell, kill it. Hey, there's a virus, kill it. So it sends that message to your immune system to come and kill specifically. And with that, you can get a rapid reduction of viral titer in your lungs that you have seen in mouse models. Yeah.
Okay.
Yeah.
So you're seeing positive results of the stuff you're testing now.
Yeah. Yeah.
So you might be eventually releasing something that will like stop the flu.
Yes. So, for example, you get a flu infection, or someone in your house maybe got a infection. Mm-hmm. So you just go to the doctor's office, and if you test positive, just take a single dose. So our drug is a single dose drug. So with just a single dose, within 24 hours, we see complete elimination of viral load from the lungs. In mice, of course.
On a small scale.
Yeah, on a small scale. But in large animal models, we are going to test, and of course, the ultimate goal is to take it into human clinical trials. Yeah.
I've always heard there's lots of different strains of influenza. So how would this drug affect? Is it for one strain? Does it work with multiple strains?
That's a very great question. So we target a protein that is called neuraminidase.
Okay.
So we are probably familiar with H1N1.
Yeah.
So these are actually proteins. So H stands for hemagglutinin and N stands for neuraminidase. And there are different types. That's why we call H1N1, H5N1, H7N9.
I've never understood that.
Yeah, I do. Yeah. Okay.
Yeah. So that's how the virus strains are nomenclatured. For example, avian influenza is H5N1. People are very scared about right now because it is very dangerous, highly pathogenic. And the seasonal influenza, we are familiar with H1N1. Mm-hmm. So so our drug, we target neuraminidase. and it binds with all types of neuraminidase. So N1, N2, N3, N4. So
Okay.
any type of neuraminidase we have tested, it binds to all of them. So it's not just for a particular flu strain, it's against multiple strains. And we have tested against the seasonal influenza strains, the pandemic strains. There was a 2009 pandemic for influenza. So our drug works against that. And also we tested against, so there are two subtypes of influenza, A and B. So it works against both. So it's you can say a universal flu drug. So not just for one particular strain.
All right, so how do you test on a person? Do you give them the flu and then give them the medicine? Are you just looking for a I'm not volunteering, by the way. Is that what you do? Have someone volunteer, hey, we're going to give you the flu, but we might cure it? Or do you just try to find someone that has it?
That's a great question. So currently how it works is once you have proven, first you have to prove that it is safe to use in humans.
Okay.
That's the phase 1 clinical trial. That's true for any drug. So any drug you develop, the first thing you have to prove is safety.
Now, how do you prove that? Do you just inject a couple people?
You just inject in healthy volunteers.
Okay.
But before you would do that, you would kind of know.
Because first you have to do a lot of toxicity and safety-related studies in animal models, and not just one animal. So that would be maybe in dogs, rats, cynomolgus monkeys, and of course mice. You have the preliminary data. So from all these animal studies, so FDA would require systematic toxicity studies, safety studies in animal models.
Okay.
So before they approve that, you can take it into humans. So of course you have to spend a lot of time, and it becomes expensive, because when you go to large animal models, those studies are very expensive. So that makes the drug discovery process an expensive one, of course. So you have to test all sorts of safety concerns, and then the FDA will give you a green signal. Okay, now you can test it in humans. And that's the phase 1 clinical trial, where you can inject your drug into healthy volunteers and to see whether it is safe.
When you first start testing, you're not looking for actually people sick. You're
just for safety.
Just we're going to side effects.
Right. And also the dose, how much drug you can actually dose, how much higher dose. That's called a dose escalation study. So what's the maximum dose you can actually inject into humans without causing any side effects?
So do you just keep raising it until somebody
there's a dose range. No.
Oh my gosh.
What the heck? You know, that's a question. People have wondered this. I'm not the only one.
So you can actually guess your safe dose range from animal studies, and from there you can see how much actually. So you start with a lower dose, and there's a dose ranging intervals, and so that you can say that from this dose to this high dose, the drug doesn't have any safety issues.
Okay.
So first you want to make sure that it is safe.
Okay.
Well, I've seen movies where, it didn't work. They just threw that body out and got another person.
Oh my god.
I didn't know if it was like the movies
I don't
or not.
want to participate in your clinical trials. That's not.
Probably the best. It's not my job.
So back to your question. So then in phase 2, there can be two parts. So in phase 2, now you want to test your efficacy, whether your drug works or not. First, you know that it's safe, but now it has to work. So for that, in case of infectious disease, people do human challenge studies. So that's very common in the field.
Human challenge.
So you infect healthy volunteers with a viral infection, and there are like standard protocols for that. So the infection won't kill them, but it's very regulated, and they will recover naturally. But we just want to see whether our drug works or not. So if alternative is we can get patients who get natural infection and then treat them, but there will be a lot of diversity, because not everyone will get the same amount of viral load. So there will be a lot of variability. So when you are just testing it for the first time, it can be a little confusing whether your drug is causing any effect or not. So that's why it's very standard to do a human challenge study. So where you can do a particular viral load to all the patients. For example, you take 20 individuals and infect them with the virus, and then treat them with the drug and see whether it works or not.
So how do you get a hold of that virus?
Are you working with, you have like samples of this in the lab?
Yes, yeah. So, for example, for infecting mice, we have mouse-adapted versions of the influenza virus that can cause lethal infection in the mice. So, because we check in mice model, we can actually do a survival study. So we can see what percentage of those animals survive. So we can give them a very high load of viral infection to see how effective our drug is.
Yeah.
But in humans, of course, people will not die due to influenza infection. Right? Yeah.
Well, I can't believe what I'm hearing right now.
So in case of humans, we mainly care about the viral load. So when you take the drug, so how soon you can get recovered from the symptoms, and how quickly it can kill all the viral infections and virus particles in your body. Yeah.
I see.
Yeah.
I imagine it costs a little bit to do that.
Yeah.
I'm guessing this is not a cheap research.
No, of course, it's very expensive. For example, the preliminary studies, when you do it in mice, that might not be very expensive. But if you go to large animal models like ferret models, so even a small study would cost you at least $50,000. So yeah, because the ferrets are expensive. They're So yeah.
What kind of place funds this type of research?
Okay, that's a very great question. So. You can get funding from National Science Foundation, NSF, National Institute of Health, NIH. NIH has a subdivision for infectious disease. And also there are government agencies like BARDA, they also fund, and you can also get funding from other voluntary organizations like Gates Foundation, Open Philanthropy, and Novo Nordisk Foundation. They actually recently they have started a PAD program, PAD initiative, that is called Pandemic Antiviral Discovery, PAD. So they are trying to find this new kind of technologies that can handle a future pandemic of influenza. So, and we also recently applied and we got our preliminary approval just today. Yeah, so it's a very lucky day.
Well, congratulations, that's wonderful.
Thank you. So now we are going to submit a full application. So yeah, like that, they are inviting applications from people who are working on these novel technologies, because if there's a next pandemic for influenza, so that we can be prepared. And we can have a drug that is tested at least in phase 1 or 2 human clinical trials. Yeah.
So is that what you're ready for at this point? Are you going to large animals, or have you already done that? You're ready to start human trials?
So now what we are doing is, till now we have been manufacturing the drug in the lab, very small scale, milligram level. But when you want to do all those toxicity studies in animals, all those safety studies, then you need a lot of materials. So now we are doing a large scale manufacturing of our drug.
Okay.
So now we have to do it in a kilogram batch or a kilogram scale, large scale. So, and after that, our next plan is to test all those toxicity and safety studies in animal models, of course in large animal models. And also we are trying to do some, so far we have tested against H1N1 and H3N2 and influenza B. So we also want to test against H5N1, which is the highly pathogenic avian influenza.
Okay.
But for that you need a biosafety level 3 laboratory. So we are actually collaborating with other partners to get that facility. And,
See, and that goes into it's like the last question I'm going to ask you, right? Because you have live virus.
Right.
Yeah.
And so do you have is it like in the movies where you have this room you go into and you get like sprayed down, and then you walk into the next room and it's like this bio chamber? I mean, is that real?
Yeah, that's real. That's biosafety level 3. So, so we don't have that here. We don't have a biosafety level 3 animal facility here at Purdue, but Professor Richard Kuhn, his lab has a biosafety level 3 lab. That's, I think, for in vitro studies where they work with live viruses and cell lines, but not for animals. So for animals, there are institutes, like we are collaborating with Institute of Antiviral Research at Utah State University. They have a biosafety level 3 lab where actually what you see in movies will be real.
That's right. I don't know if it's just me.
I know.
I need to know these things.
Yeah.
Well, this is exciting.
Yes, it is.
And awesome.
Yeah.
And informative.
Yeah,
We appreciate this.
Yeah. Thank you so
for your
much.
time.
Thank you.
Thank you for coming and chatting with us.
Thank you so much.
Thank you for listening to this episode of Science from the Experts from Purdue University Superheroes of Science.
If you like this episode, subscribe, give us a positive review, and share the love. Boiler up!
Hammer down.